वक्रतुण्ड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देवा सर्वकार्येषु सर्वदा। वक्रतुण्ड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देवा सर्वकार्येषु सर्वदा।
वक्रतुण्ड महाकाय सूर्यकोटि समप्रभा निर्विघ्नं कुरु मे देवा सर्वकार्येषु सर्वदा।
लम्बोदर नमस्तुभ्यं सततं मोदकं प्रिय निर्विघ्नं कुरु मे देवा सर्वकार्येषु सर्वदा।
लम्बोदर नमस्तुभ्यं सततं मोदकं प्रिय निर्विघ्नं कुरु मे देवा सर्वकार्येषु सर्वदा।
I would request Dr. K.D. Nayyar sir, our guest of honor, to say a few words about this meeting. Dr. K.D. Nayyar sir.
Good morning.
Sir, before you start, sir, may I have the honor of inviting you personally? I think he's the most celebrated IVF specialist of our country. Sir, we can't see you. You are hidden behind the camera as always. Dr. K.D. Nayyar is ASRM star, and he has got, I think, the most celebrated IVF specialist from the side of ASRM and SH, Dr. K.D. Nayyar. He's got a large number of publications, large number of poster presentations, and he's trend-setting in research from India. So thank you so much, sir, for blessing us, and we are thankful to you for joining us early in the morning, sir.
Thank you, Dr. Pankaj. Good morning, everybody. I must congratulate Dr. Pankaj Talwar and the whole team to bring this focused one-day certified online course on genetics, the journey into genetic makeup from basic to advanced. To be there to guide this course throughout the day. And I'm very sure that all the delegates will be enriched with clear take-home messages and to gain knowledge about this complex specialty. This is a complex specialty, that is why I say you need a very clear messages. And once again, I wish this course to be a benchmark success so that it can be a regular feature in the future. Thank you, Dr. Pankaj.
Thank you, sir.
Thank you, sir. Thank you so much for giving us your blessings. I would invite ma'am, Dr. Gauri Devi ma'am, to give us her blessings and say a few words, please.
Before Dr. Gauri comes, good morning, madam. Aap kahan hain? I can't.
Morning, ma'am.
You can't see me?
I can't.
No,
see.
ma'am.
Yeah, I can. No,
We
because-
can now.
So Dr. Gauri Devi is here, and all of you who doesn't know Dr. Gauri Devi, she's a past president of Indian Fertility Society, my mentor, my guide. I think in case you talk about complete, the complete cohesion between the fertility and obstetrics, she's the one. There is no VIP in Delhi NCR whom she has not delivered. And actually, I'm speaking from my heart. Look at any VIP, anyone who's doing good, or even you go to North Delhi side. I think every baby has been delivered by Dr. Gauri Devi only. Dr. Gauri Devi believes in cohesion and integration, and she's a master IVF specialist and does a lot of work in totality. She's got a big hospital in North Delhi, and she's working at Fortis Hospital, Delhi also. Thank you so much, ma'am, for joining us and enriching us.
Thank you, Dr. Pankaj, for the nice introduction. I don't have a very big hospital. Anyway, see, human genetics is developing very fast. 20th century has been really good in the fast progress of genetics. And it is very difficult for clinicians to catch up with the development. And it is very necessary to have such CMEs as being conducted by Dr. Pankaj Talwar so that people understand what is genetics from the basic to the clinician to what the latest development are. Now, this development of genetics, it also leads to pharmacogenomics. If you understand the genomes, it can also help us to identify inherited disorders. Maybe in the future, with gene editing, etc., we might be able to correct some of these monogene disorders, though it is only in the developing stage and developing stage here, because many ethical, social, and moral reasons are there, in which in plant biology, animal biology, all this are going very fast. But when it comes to human, ethical, moral considerations come, and we have to take a lot of permissions and licenses before we can go into that. But anyway, with genetic studies, in the present scenario, we have been able to understand infertility better, some of the recurrent pregnancy losses, some of the recurrent implantation failures, and we can also, with PGT, AM, etc., we can prevent some of the diseases and save the babies from having anomalies. So let us together see this, listen to this CM from morning 8 to 4 o'clock. And congrats, Dr. Pankaj, for holding such a focused CME on genetics. And as Dr. KD said, we should have such CMEs more often. And wishing all the best, and to all the delegates, I hope you all will go with a lot of information today at 4 p.m. Best of luck.
Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am. Thank you, ma'am, for your blessings for us. I would request Dr. B.C. Das, Chairman MIT, to please give us a few words of wisdom. Dr. B.C. Das, sir. Sir, good morning.
Good morning, sir. So,
Good
Dr.
morning,
Das, sir, before you start, sir, let me have the honor of inviting
Good morning.
you.
Good morning to all, and Dr. Kedina, Dr. Gauri Devi, Dr. Talwar, and also the organizing team, Dr. Nisha Bhatnagar, and her colleagues, and everybody who have joined, my colleagues also. As Dr. Pankaj Talwar has initially mentioned, that we have a strong or intimate collaborations with the Indian Fertility Society, of which the present president is Dr. Talwar. But at the outset, I congratulate Professor Talwar for organizing such an illuminating workshop on genetics, that is from basic to the advanced. But that's very interesting and very important for the successful IVF practitioners. They need to know the genetics, the genetic abnormalities when you deal with the gametes and before the fertilization and all the implantation. So this is an illuminating workshop that you have accumulated. There are good speakers from the basic to molecule to next generation artificial intelligence or digital health that can help and to solve our problem, to understand the genetic intricacies of this, any problem or any defects or abnormalities occurs during this the IVF procedure. But not only that, in general, the knowledge of the genetics, from the basic genetics to advanced molecular genetics and beyond, is very important. So I think it's an excellent, but I would appreciate that what you have started. I have no idea that you have, before you had any such meeting of the on basically specifically on genetics. So, but if it is continued once in a year, one day, one can make it convenient to have this. If not 6 monthly, but at least yearly, you can have this, so that we can easily know what is the new in the field that Dr. Gauradevi has mentioned, the pharmacogenomics. So that's very important, pharmacogenetics to pharmacogenomics. So now the cytogenetics or cell genetics has become cytogenomics. So from the genetics to genomics, what is that? We have, that means the genetics to the simple cytogenetics to molecular genetics or the molecular genomics. So these are very important to understand because science is every day changing. So if we are working with the scientific or this field, and that too most important is that we are working with the human life, that we are giving a new life to or new human being to come into this world. So through this procedure, so we should be very careful that we are 100% perfect to it. So there are many other intricacies, but certainly genetics or abnormalities to understand before we go for this IVF. So that's very important, and genetics is certainly very important, which can be that the technology is improving day by day, so that we have to be illuminated or we have to be knowledgeable. Through this, everybody may not have the opportunity to go through, but if you have this type of meeting, it will really give a boost to this area of specializations. So thank you so much for giving an opportunity to talk in this occasion. Thank you. I hope this meeting become very fruitful. I'm sure the type of speaker you have accumulated, I'm sure, and I see a lot of participants' enthusiasm, and I think you will not be disappointed as I see the list of participants and the topic covered. So thank you and all the best wishes for the successful meeting. Thank you very much.
Thank you very much, sir. Thank you. And I think we have already got around 100 people who have joined in beginning of the day. So I think big clap for Nisha and Meenu for making it possible. It wasn't possible without your support, and definitely in the backend team, I've got Mohit who has been running the whole show. I've got Ashna, Sandhya. So everyone, all the girls, good luck, best of luck. And as I pass on the mic, we're late by around 5 minutes. I'll like to just say hello to Professor Subhrajit Biswas from MIT. I can see him there in his pink shirt. Thank you so much, sir, for joining it so early. I can see Puneet Rana. She's also here. She's been helping us in this coordination. So with this, with heart full of gratitude and thankfulness, I hand over the mic to Nisha Bhatnagar for the further proceedings. Thank you.
Thank you, sir. Thank you, sir. Thank you, Dr. B.C. Das, sir, for giving us your words of wisdom and your blessings. I would, with sir's blessing, and I am really thankful to Dr. Pankaj Talwar, sir, for giving me this opportunity. I have, like, if we talk about the beginning, I am at ABCD stage and I have to learn writing the book from sir. So all his guidance always needed. And with sir's blessings and all of my seniors' blessing, we begin the day. Dr. Meenu, can we invite the first speaker, please?
Absolutely, ma'am. I take the honor of inviting Dr. Professor Subhrajit Biswas, who is the first speaker for the day. And the topic is structure of DNA coding and transcription. So, sir, can I have the, can I see?
The CV is there on the...
So, sir is the director of MIT University of Molecular Medicine and Stem Cell Research at MIT University. He's former faculty member, Institute of Liver and Biliary Sciences, New Delhi, principal investigator, leukemia scheme from LTMT, National Cancer Institute, London, UK, scientist at the Vanderbilt University Medical Center, USA. He has done his postdoctoral fellowship from Medical University of South Carolina, Charleston, USA. And he has been awarded many awards like the Lady Tata Memorial Trust Award 2010-2011, award in recognition of excellence in cancer research from VUMC USA. And sir's special interests are working in cancer, immunology, checkpoint pathways, miRNA therapeutics, very interesting, sir, and liver diseases. So we welcome you, sir, and would please start the day with the first talk from you, sir.
Thank you very much for your kind introduction. So, yes, I'm the first speaker, and I was designated for giving the talk on the DNA structure. So, can I share my slide? Because I think there is an audience over there who keenly waiting for the understand the DNA structure,
Yes,
transcription, and coding.
Mohit, you will need to stop sharing.
Ma'am, so you can share your screens.
Yeah, it's coming now, right?
Yes, sir, we can. Yes, sir, we can see.
Yeah, in the full, yes.
Sir, you
So,
need yes.
yes. So I'm directly starting with the DNA structure. So as mentioned by the panelist over there, and Professor B.C. Das sir, Pankaj Talwar sir, and this is the DNA is a unique structure which allow it to be the hereditary molecules and allow it to store the instruction for detecting the cell activities. So very important things. So I am finishing within half an hour. So I'll go through the some of the basic things, how we understand the structure of the DNA, incorporating the three component of the nucleotides. What are the outcome of this learning to the half an hours? Do we want to know about the constructed DNA molecule and how it show the complementary base pairing, how we can label the different parts of the DNA molecules, and how we can predict how the DNA replication and transcription is going on. So DNA has to be able to store the hereditary information in some sort of code. So that is the subject and be easily and accurately copied as copied in past and every time in the whole the cell divides. That the major important things, what properties the DNA allowed to do so. So in this context, I want to introduce the two big scientists got a Nobel Prize in 1962. It's named James Watson and the Crick and who developed the big institute in USA, Cold Spring Harbor Laboratory. And another two guys' name also came, the Wilkinson and the Franklin. So this is the history of DNA structure and the consequence of that. So if we think about the simple, what is the DNA? DNA stand for a deoxyribonucleic acid. It is a long molecule makeup of the monomer called the nucleotides, and nucleotides made up of the three parts, a carbon, sugar, a phosphate group, and a nitrogenous base. So that is the chemistry part all of you know. So this is the DNA structure. This I'll going through the slides. The DNA often described as a double helix, all of you knew. And what is the twisted ladder? So this is the very interesting things I want to share with all of you guys. I understand that is the all the structure and with the chemical the bonding, it is difficult to remember at a times. But this is the sugar base and the nucleotide, the structure is very interesting. So this is, if you look at the backbone of the DNA, is formed in the alternating the sugar and phosphate held together by the strong bond. So what is this bond? So I want to mention that is the interesting bond is the hydrogen bonding. That is the Watson and Crick also didn't know about that time. And the which runs the ladder are formed by the four nitrogenous base and held together by weak hydrogen bonds. So if you think the box one, that is what form of the backbone of the DNA, where are the weak bonds in the DNA, where are the strong bonds of the DNA, and why do we think is the DNA is very important? So this is the major nitrogenous base which contain the DNA. Four nitrogenous base is called adenine. It stands for A. Thymine is T. Cytosine is C, and guanine is the G. So basic base pair rule, if we look at that, so the predictive way, that is A always pair with T and C always pair with G, that is we know. So I'll come up with the detailed structure about that. So DNA is said to be complementary. Each strand can be used as a template for other half using the base pairing rules. So if we practice that way, that is one strand of the DNA has a base structure of the TACGA, So we can think about that is a complementary strand will be the ATGCTAA, just the complementary strand. So if we practice like that, so we can do that. So what determine the message that DNA contains? What is the base pairing rules of the DNA says? What about the bond each other? And if half of the DNA reads the ATCG, what will be the order of the letter of the strand? And how it will go into the translational level when the DNA will be transcribed to the mRNA, then mRNA will go to the making the peptides and peptide ultimately to the the proteins. So as I have mentioned that DNA is made up of the nucleotide, which are made of the 5-carbon sugar, phosphate, and a base, and there is a 4 different bases in the DNA molecule: adenine, cytosine, guanine, and thymine. So what are these adenine, guanine, and cytosine and thymine? So there is a purine ring and pyrimidine. So easy to remember, that is adenine and guanine are the purine, and cytosine and thymine are the pyrimidine bond. So that experiment long back done by the few scientists is very well known, the Griffith, Hershey, Chase, and finally this is Rosalind Franklin's Nature paper. So DNA molecule which hold the genetic information, and DNA is a polymer which made of the monomer, and it's the hydrogen bonding is the important parameter to make the DNA structures and helical level. So this is the complementary pair, that is adenine and thymine have 2 hydrogen bond. You can see over here. So I can take one. Yeah, I'm using the marker. And the guanine and cytosine has a 3 hydrogen bonding. So there is it's called complementary pairing of the 2 double strand. So purine and pyrimidine are the heterocyclic base, and there are the 5 common bases of the nucleic acid. The note that GTU exists in the keto form and not the enol form found in the phenols. And nucleosides is the glycosides of a purine or the pyrimidine, heterocyclic base, and the carbohydrates. The C-N bond involve the aromatic carbons of the carbohydrates, and the carbohydrates for the nucleic acid are the D-ribose and deoxyribose. I will tell about that is 2 kind of the pentose sugar. One is D-ribose and deoxyribose. So simply, if we gone for the competitive exam and the understanding of the nucleoside and the nucleotide. So what is the nucleoside? Nucleoside is a carbohydrate plus base, that is ribonucleoside or 2-deoxyribonucleoside. So if you look at the nucleoside, so if this is a structure of the adenine, when it's add up with the pentose sugar, it is coming as adenosine. It's a nucleoside. And it can be guanosine, and it is also nucleoside. It can be cytidine, or it is thymidine or uridine. So to differentiate the atom of the carbohydrate from the base, the position, the number of the carbohydrate is called as followed as a prime. The stereochemistry of the glycosidic bond found in the nucleic acid is the beta. So what is the nucleotide? So this is the basic, very interesting question. Always, if you give the exam for the GATE, NET, and the other competitive exam, it is a common question: what is the difference between the nucleotide and a nucleoside? The nucleotides is a phosphoric acid ester of the nucleoside. That means nucleotide made up of the nucleoside plus 1 phosphate group. So ribonucleoside mean that it may be ribonucleoside which is present in the RNA, and deoxyribonucleoside which is present in the DNA. So ribonucleoside 5 monophosphate, you can see the structure over there, or it may be deoxyribonucleotide. So this is the just a example. So what is the major enzyme play to catalyze the phosphoryl transport reactions from the ATP to acceptor of the substance? So it's not a thermodynamic class, so I'll not go into the details of the how ATP is the playing role in the adding of the phosphate groups with the nucleotide. So this is the enzyme is named common enzyme I have been taking. It's a kinase enzyme. And definitely, you would have to read about the bioenergetics and ATP and biogenetics and the more understanding of this pathway. So I can mention one very famous guy. Name is the Earl Sutherland, who got the 1971 Nobel Prize in Medicine and Physiology, who discovered the how the phosphoryl group. So it's a big science, and basic science is very important to understand the DNA structure and the long term. Though it's started from the 1960, now we are in 2024. It's a long way we are they know about the all about the DNA structure. So phosphodiester nucleotides and the polynucleotides are the chemical linkage between the nucleotide units, and the nucleic acid is the phosphodiester bond, which contain the 5-hydroxyl group of the one nucleotide you can see over here, and one is 3-hydroxyl group of the next nucleotide. So the way the DNA structure or backbone actually going to make. And the nucleic acid is negatively charged. And if you look at the discovery of the nucleic acid structure, that is Avery, MacLeod, and McCarthy, is the strong evidence of the DNA is the genetic material, and which is found in the nucleus in the eukaryotic cells. So another guy name is the Chargaff, who made the rules, that is careful analysis of the DNA from a wide variety of the organisms which contain the adenine, thymine, cytosine, and guanine, and which varied accordingly to the organism. So however, Chargaffs always follow that is ATA is the link with the P, and C is linked with the G, but he also not that time much of the sure about the hydrogen bonding that time. So Watson and Crick structure, which is Nobel Prize, I already mentioned in 1962 with the Wilkins, and they discovered the how its secondary structure of the DNA or the double helix. So initial, it is a like with like, that is parallel helix, and does not fit with the Chargaff's rule, that is A only bind with the G as T, and G is binding with the equivalently with the C. So it's not. So you can see here that is purine, if we look at the structure, and pyrimidine, that is the, we can, that time it is called the wrong tautomer. Then the Watson is the discovered that is double helix. So I can remember when I was actually in the student in the BSc class, in the long graduation, that was the confusion was that, and one of our teachers came, and he made a interesting story in the classroom. That is the how that is the discovery of the Watson tree, and they are trying to find out the exactly what is the length of the DNA, and the crystallography is not matching at all. So at the time they twisted their own paper and threw in the trash bag. Then the idea came that is there is a something is that the hydrogen bonding between the two nuclear the two nitrogen base is something different. So they able to find out that is the double helix is the twisted one. That is one helical turn is coming around 34 angstrom. That means that is if it is the DNA helix is the twisted one, so one will be the major groups and one will be the minor groups. So I'll coming in the more about the DNA backbone. So this is the computer generated model. And if you look at the tertiary structure of the supercoil of the DNA, the each cell and contain about 2 meters of the DNA, and DNA is packaged by the coiling around the core of the protein is called the histones. The DNA histone assembly is called the nucleosome, and Professor B.C. Dasar will describe the more about the chromosome nucleosome structure along with the histone, which lysine histone is always rich in lysine and adenine residue. So the central dogma, what of the Watson and Crick found at it, that is when the DNA, the genome is one side is replicating for the proliferation of the cells, and the another side the DNA is coming out and making the mRNA, that is called transcription, and the product is called transcriptome, and finally that is mRNA when translated into the protein, that is a proteome. So expression and transfer of the genetic information in terms of the replication, transcription, and translation will call the central dogma. And the replication is the process by which DNA is copied with a very high fidelity, that means without error. And transcription is a process by which DNA genetic code is read and the transfer into the messenger RNA. And this is the intermediate step in the protein expression. The process by which genetic code is converted to the protein, the end product is of the gene expression. So DNA sequence code for the RNA sequence, which codes for the protein sequence. That is the central dogma and the beauty of the inheritance of genetics. So DNA replicated by the coordinated effort of the number of the proteins and enzyme, and the for replication, DNA must be unlocked first, then uncoiled, and double helix would be unwound. So there is no need to remember everything in the simple half an hour classes, but these are the major enzyme is the replication for the important for the DNA replication is topoisomerase, helicase, DNA polymerase, DNA ligase, and topoisomerase is responsible for unknot and uncoil the DNA. Helicase, this protein which unwind the DNA double helix, and the DNA polymerase, the enzyme that replicates the DNA using each strand as a template for newly synthesized strand. And the DNA ligase is an enzyme that catalyzes the formation of the phosphodiester bond between the pieces of the protein. So DNA replication is a semi-conservative and each any questions? Okay. So DNA replication is a semi-conservative. That is, each new strand of the DNA contain one parental strand and one is a template one strand. And this template one strand is called one newly synthesized or daughter strand. So if you look at the picture where the unwinding of the DNA by the helicase, which expose the DNA bases, that is a replication fork, so the replication can take place. And helicase, the hydrolyzes the ATP in order to break the hydrogen bond between the DNA strands. So DNA replication, if you look at this, this is the DNA strand. One is red, one is 3' to 3' based on the phosphate group, and another is a blue one is going to the 5' to 3'. And if the fork is opening from this side, that means DNA strand is replicating from 5' to 3' order. So this is the major order of the DNA replication, and DNA polymerase, which is major enzyme for the replications, will bring the unwinding and the replication process through 5' to 3'. So what is the DNA polymerase? It's the newly strand replicated from the 5', the start from the 3 end of the template, and the DNA polymerase are the magnesium ion dependent, and the deoxynucleotide 5-triphosphate is the reagent for the nucleotide incorporations. So you can see here that is a phosphate group, how this magnesium, this is the strand, and the DNTP is coming and it is binding with here, and the new strand is forming. So 3-hydroxyl group of the growing DNA strand act as a nucleophile and attract the alpha phosphorus atom of the DNTP. So make it easy, the replication is a leading strand occur continuously in the 5' to 3' direction, but on the other hand, when I am mentioning that is DNA polymerase has a restriction and the limitation to move only in the 5' to 3' direction. So then there is issues with the another strand, how the DNA polymerase will move. So DNA polymerase has to move the opposite direction. So then there should be a fragmentation of the DNA synthesis occur, and where the lagging strand occur discontinuously. So that is why it's called semi-conservative. So short DNA fragments are initially synthesized, you can see here, and then the ligated together. Of course, there is an initially involvement of the RNA primer, and where the DNA replication occur, the RNA will synthesize small fragment of the complementary strand. Then the DNA polymerase will bind over here and move to the 5' to 3'. And though the replication fork is going towards the 5' to 3' one side, so one strand will be continuously replication and one strand will be discontinuation of the replication. So the scientist name is Okazaki, who's discovered this discontinuation bank by the molecular biology technique, and that's why his name is, and the strand is called Okazaki strand. So DNA replication occur by high fidelity. The most DNA polymerases have high intrinsic fidelity, and many polymerases have the proofreading property. And repair process seek out, and the repair base pair mismatch due to the unfaithful replication. And this is also kind of the big chapter to understand the DNA replication process. So honestly, in the US, first time when I went for the postdoctoral study, I work on the DNA replication lab in South Carolina. And so I have done by hand and the bench work with the how the replications occur. So that is the collision between the replication and transcription process. So anyway, back into the subject, that is what is the ribonucleic acid. ribonucleic acid is a RNA contain the ribose, then deoxyribose, and uracil rather than thymine. That is the difference between the DNA and the RNA. So there are the three kinds of the RNAs. One is called the messenger RNA, and the another is a ribosomal RNA, and then the transport RNA. DNA is found in the cell nucleus, and mitochondria. RNA is a more dispair in the cells. So what is the transcription is only one of the DNA strand is copied, that is coding the antisense strand, and RNA polymerase replicate the DNA sequence into the complementary sequence of the mRNA. And mRNA are transported from the nucleus to cytoplasm, where they act as a template for the protein biosynthesis, that is called the translation. And three base segment of the mRNA, which codes for the amino acid, is called the codon. The reading frame of the codon is defined in the start and the stop codon. I'll tell a little bit brief about the codon. And this is the standard genetic code, and there is a different amino acid that codes for the three codes of the nucleotide. So if you look at this structure, that is the mRNA structure, one side is 5' cap and one side is the 3' UTR region, and there is a codon for the start, and the mRNA is positioned to the ribosome through the complementary pairing of 5' untranslated region of the mRNA with the rRNA. Then the transport RNA, which carries the amino acid on the 3-terminal hydroxyl, it is called the amino acyclyl tRNA, and ribosome catalyze the amino bonds formations. So what is the ribosome? It's a large assembly of the proteins, and rRNA that catalyze the proteins and peptide biosynthesis using the specific complementary antiparallel pairing interaction between the mRNA and anticodal loop of the specific RNAs. So although the single-stranded, the complementary sequence within the tRNA that gives the definite codon. So if you look at the simple structure of the amino acyl tRNA, you can see that is T side, C loop, D loop, and the variable loop. So this is the tertiary structure of the tRNA, and the three base codon sequence and the mRNA and complementary to the anticodal loops of the appropriate tRNA. The base pairing between the mRNA and tRNA position in the amino acid transport to the growing peptide chain. So this is the very brief about the translational or protein synthesis process, and there is a two sides of the ribosome, what is called P sites and A sites, where the tRNA will bring the amino acids over here, and when the mRNA is sliding over the ribosome complex, each amino acid is attaching over there in the E side, that is elongation side, and ultimately the formations of the proteins. So let us a little discuss about little details, because maybe I have another 10 minutes times. That is how the DNA sequencing we can do. So nowadays there is a automatic NGS new generation sequence already started. So if you want to know about something about basics of the DNA sequence, you must know about the some scientists who discovered that is how we can sequence the DNA. That is how we know about our genetic information, what is the information and what is the sequence. So first name came with the Maxam and Gilbert, who relies on the reagents that react with the specific DNA base that can subsequently give rise to the sequence with the specific cleavage of the DNA. And the second name came, and they're very famous for a long time, the sequence we used to do by the Sanger method. And its method is based on the enzymatic replication of the DNA fragment to be sequenced with a DNA polymerase with help of the magnesium and the deoxynucleotide triphosphate. So this is the new concept bring by the scientist Sanger, and which truncated the DNA replication. So I'll give very brief about that. And before that, we need to know about what is the restriction endonuclease. Actually, that particular enzyme found in the bacteria. And those bacterial enzymes that keep the DNA in the particular specific sequence. So if you can remember few of them like EQR1, BAMH1, means they clip the DNA sequence in one of the specific sequence code. That is, if it is a GAATAC and complementary sequence is CTTAG, then the equal when cut exactly in the 5 prime in between the G and A, this is phosphorus group, and in the another side that is a 5 prime to 3 prime, another region between G and A. So that is the way the restriction endonuclease or the equal one. So this is the basic concept. And what is the deoxynucleotide triphosphate? And actually, it's a lack of crucial hydroxyl group which is present in the DNTPs. Now, if we run the DNA sequencing in the test tube, that is, we'll add first the different kind of the all ATP, GTP, CTP, and the TTP. Then along with that, we are adding one the reagent we are keeping, that is DNTPs. So in DNTPs doesn't have the crucial hydroxyl group, and due to this crucial hydroxyl group, when the DNTP is incorporated into the elongation of the primer to be, then the DNA replication will be terminated. The DNTP is specifically incorporate the opposite its complementary nucleotide base. So if the DNTPs and all ATP are, if somehow we tagged with the radioisotope and other tagging, so we can in the gel electrophoresis process, we can find out the particular that is different length of the sequence if it is a terminated into the G. That means each sequence, the panel of the gel electrophoresis, one panel we are adding DDA, one panel we are adding DDG, DDC, and DTT. So if when the DNA replication will occurring in the tube, there will be the particular place where the DAA adenine is there will be termination. So you will get a number of the different size of the fragments, which is separated from the larger fragment to smaller fragment, and ultimately by radioactivity exposure, you can see the particular the sequence of this DNA. So that is the oldest method so far. Nowadays, that is a next generation sequencing already came, and we directly by the method we can directly we can see the DNA sequencing. So there is a huge genome project already running. And when we are doing the PhD, this is I was telling in the 2000, 2005, that time that is the human projects come and it took 10 years to complete the total human genome code. And there is a big news in the Nature and Science. But nowadays, the total human genome we can do within 4 hours, 3 hours, and that is the beauty of the science now. So far that is in the when we are talking about the laboratory. So how we can the qualify or amplify our the DNA in the laboratory. So that is the process is called the PCR, polymerase chain reaction. That is where we use the heat stable DNA polymerase. It can be tag polymerase, which is found from the thermophilic bacteria, or few that is the geothermic vent bacteria, that is vent polymer is also important. And with the help of the magnesium and the primary strand and the DNA template, we can in the laboratory we can make the amplification of the DNA. So that is the Carey Mullis in
Sir, a gentle
1990.
reminder for the time, sir.
Yes,
Yes.
when it will be finished
Right.
within 5 minutes?
Yes, sir. Yes. Please continue, sir.
5 minutes?
Yes, sir.
Okay. Yes,
Please
I am
continue.
almost finished. Yes. So this is the polymerase chain reaction. That is a first step is the denaturation, then annealing, then extension, and this is the I'm just finishing with the DNA structure and transcription translation. I just brief, and the in the half an hour is not enough. I understand. That is just telling about the James and Watson. That is a discovery part, and which is the unknown story, and it's all not too I could not say that is unknown. Different news channel and already people know about that is the person named the Rosalind Franklin, who died in 1958. Actually, her X-ray diffraction pattern of the DNA actually big block for the Nobel Prize oriented. And this is the one later form, the James and Francis. That is, the nucleotide has a 3 basic part carbon in the first declare. And here you can see that is they have taken the Rosalind Francis photographs was very helpful to us. And here at the peak, actually, ultimately, the what is happens in case of the discovery of the Watson Crick. We can go in here, the puzzle. And this is, I'm showing the Cold Spring Harbor Laboratory, some of the pictures. So in that time, there is a major confusion between the DNA helical structure came with the hydrogen bonding. And you can see here that is a pairs of the 2 hydrogen bonding between the 2 nitrogenous base. And there is a concept was the Hookesten pairs and the reverse Hookesten pairs. Actually, when they are chemically synthesizing and trying to pairing the nitrogenous base along with the backbone, the calculation was exactly not matching with the things. So ultimately, the results came from the crystallographic structure, which is done by the Rosalind Franklin. So what is crystallography? Just one line. That is, if you do the crystals of any powder or crystals, though the helical fibers give this kind of the diffraction pattern of the crystallography. What was found by the Rosalind Franklin? Ultimately, the Watson and Crick indirectly obtained the republication version of the Franklin's DNA extra diffraction data, and possibly without her knowledge, and the republication manuscript is Pauling and Coley given the critical insight of the DNA structure. And that Nature paper actually brings this to the person in the Nobel Prize, that is the 2%, and share with the 3rd person is Wilkinson. But in the history, that the Rosalind Franklin was not Nobel laureate, and that is the tragedy of this whole story. But people know about that 2062 Nobel Prize of the Physiology of Medicine, and what about the Rosalind Franklin? So that is the I am just showing some of the pictures. That is a long back Watson Crick, and if you go to the Cold Spring Harbor Laboratory, you will find the original structure and their research papers, as well as the whole story about that. So this is nowadays that the DNA structure, we know about it, we know about the transcription and translation. So thank you very much for the lack of time. I have to finish because next speaker, maybe Dr. Pallavi Agrawal, will continue with the inheritance and the epigenetics part of this, the replication, translation, and translation process, centrally the central dogma. Thank you very much. Is any
Dr. Meenu?
Thank you so much, sir. Am I visible and audible, ma'am?
Yes, yes, you are.
Okay. So thank you so much, sir, for your wonderful talk. It was really very exhaustive and informative.